Well, guys, we did it. You made it through all of the population genetics, all the underlying principles of evolution, natural selection, in order to get here where we begin our survey of all of life on the entire planet at the end. We're going to talk about, starting today, we're going to talk about life from every different possible domain. Today we're covering domain bacteria and domain archaea, and then through the rest of the course we're going to be covering domain eukaryota. So all of the more complex life, or what we would call complex life. There's a lot of diversity just within these two domains, though, bacteria and archaea, and that's what we're getting into. It's important to remember that prokaryotes are paraphyletic. It's that term that you guys hate, paraphylogeny. Para meaning almost but not quite. So this is not a monophylogeny. A monophylogeny would be from that last universal common ancestor and include all of the descendants. We have a common ancestor and some, but not all of the descendants. We're not looking at this group over here, the eukarya. We're looking at bacteria and archaeans, which don't include that other group. So this is paraphyletic. Archaeans, one of the two domains, are most closely related to eukaryotes. We have a lot of similarity with archaeans. But you'll notice, based on this image, that basically archaeans and bacteria split from that last universal common ancestor into archaean bacteria. And then as they evolve, eventually they come back together to make eukaryotes. What is it when you're sending genes? It's not a direct lineage of genes, but you're crossing them back and forth to another species. What kind of gene transfer is that? When you're going sideways. It's called horizontal gene transfer. There are an estimated, lots of bacteria. What is that number? Okay, 1 million, billion, right, the billion's there, trillion, quadrillion, pentillion, hexillion, septillion, nonillion, octillion, an octillion. Lots and lots and lots and lots of bacteria on the Earth at any given point. Literally, we estimate half of all the biomass on Earth is made of bacteria. A lot of your biomass, a measurable amount of your biomass, is bacteria. In a pinch of soil, do you guys know what the term pinch means? Literally, it's like pinch. Yeah, pinch. A pinch of soil. I know it's the weirdest when you're have a recipe and you got a pinch of soil. That's the weirdest term ever. Well, okay, it would be really weird if you put a pinch of soil into a recipe. But like a pinch of salt. But if you think about a pinch of soil has as many prokaryotes as there are humans in the entire world. That's a lot of bacteria when you think about it, especially when you think like kids eat soil, because kids aren't smart. These prokaryotes will live in every single possible environment. Wherever we have looked and found life, we have found prokaryotic life. Everywhere, from frozen remotes to the bottom of the sea to acid lakes. There are prokaryotes everywhere. A lot of times people will say that bacteria and archaeans are the oldest organisms, but it doesn't hold true, because if you think of that phylogeny I gave you, they all stem from the last universal common ancestors. Our lineage has evolved for just as long as whatever E. coli lineage there is. This, by the way, is a picture of seagrasses. There are seagrasses near Australia that are thought to the seagrass bed, because they are cloned of each other's, that are thought to have predated the last ice age, of the same basic organism. Over the billions of years that bacteria and archaea have been around, they've established a whole lot of metabolic diversity. That is, if there's something out there that can be broken down, they will have broken down it. They can devour just about any chemical you can think of, and can, one lineage anyway, if there's a resource they can steal, if there's an electron they can take, they will. This is likely because they have a very, very rapid reproduction rate. So they have a high mutation rate combined with a high reproduction rate. That's going to increase, as we recall from a previous lecture, speciation. That is, there are going to be differences or accumulations and differences in these organisms very, very, very rapidly. There is rapid evolution in bacteria. So we've got two broad prokaryotic groups, archaeans and bacteria. And I want to get into some of the differences between them. One major difference between archaeans and bacteria are that archaeans have histones. Your chromosomes are built by taking DNA, eukaryote, your DNA, and wrapping it around a protein called a histone. Then it goes on to another histone. And this is sort of what gets them condensed down into those forms that you guys look at under microscopes when you're studying mitosis. Archaeans also have histones, but bacteria do not. Archaeans have a few extra ribosomal proteins. So what we're looking at is basically we've got a this one on the far right side is a bacterial ribosome. Here in the center we've got an archaean ribosome, and on the far edge we have a eukaryotic ribosome. And you can see the prokaryotic ribosome and the archaean ribosome are very, very similar. It's because of this difference in ribosomes that when you take an antibiotic, it will kill the bacteria but not you, because usually they target that ribosome. If your ribosome doesn't work, what stops happening? If you don't have ribosomes, what do you also not have? Proteins. And proteins do everything. You've stopped protein synthesis if you could stop that ribosome. Your ribosomes have some additional structures that make them sort of immune to antibiotics. So do archaeans. Yes?
Did you say that the archaeans share the histones with eukaryotes as well?
Yes, so archaeans and eukaryotes have histones. Archaeans and eukaryotes have similar ribosomes. Their RNA polymerase looks just like the eukaryotes, and I know you guys don't want to look at chemical formulas and the structure of molecules. Basically, what I want to point out is we've got at the bottom here a bacteria, an archaean, and a eukaryote. You can see that the archaean and the eukaryote look similar. They've both got this big purple structure sticking off the top. The bacterium does not. That means that some things that impact bacteria do not impact archaeans and eukaryotes. More chemistry for you. Archaeans have what are called ether-bonded phospholipids instead of ester-bonded phospholipids. And here's the difference. Have an ether bond. Ether bonds have cross-linkages. So phospholipid, then you've got the tails of the phospholipids. If you have an ether bond between the phospholipid legs, they're holding on to each other. Typically, in a normal eukaryotic or bacteria cell, you have ester linkages, which means there's nothing that connects the tails to each other. Here, the tails are holding on tight. You're holding on not just to your head, but to the tail next to you. And then you're holding on all over the place. It creates stronger bonds. That means those cell membranes aren't going to fall apart as easily. Okay, the way I always put this, when you guys were younger, did you ever have to like build a fort out of popsicle sticks for a school project? You never had to do that? That's like the most, okay. Have you ever done any, okay, then I'll just explain this. Bear with me. Imagine, if you will, trying to make a wall of popsicle sticks. You take the popsicle sticks, you lay them next to each other. How stable is that wall going to be? It's not even a wall, right? Okay, let's sit them up and you put them next to each other. The way you would get them to stay together is you'd put a cross brace across the back. Does that make sense? Think of a normal fence. You put a brace across the back. You don't just stick sheets of wood in the ground. Your cells and bacterial cells are the equivalent of these ester links that are just boards stuck in the ground. But archaeans have ether linkages, which gives them those cross braces to hold everything even tighter. That makes their membranes hold together in conditions that ours will fly apart in. Yes.
Do bacteria have ether or ester, or neither?
Bacteria have the same as us, which I think I just said ether. Ether? Ester. Yeah. If we're ester, bacteria are also ester. We get our phospholipids from bacteria.
Okay.
We get our DNA basically from archaeans. Because of these cross linkages, archaeans are going to be considered what we call extremophiles. They live in the most extreme environments out there because nothing else can. You have types. I am not going to ask you to memorize the different types. Here's the question I will ask you on the whatever test is coming up. Give 2 examples of environments that archaeans can live in. So you don't need to memorize everything. You might be an acidophile. They live in, as the name might suggest, acid. Very low pH levels. This is a lake in, I want to say, Java. And you'll notice that these weird areas are usually near geologically active places. So here's a volcano. This lake is the most acidic lake in the world. It is light blue because nothing except archaeans can survive in it. Say it again. The lake is in Java, Indonesia. This right here. So see this blue area off to the right. Sorry, it's the closest I could get was a satellite view. You could be an alkalophile. These grow at a pH of about 9 or above. So where you had acidophiles, now you have alkalophiles. We're very basic areas. Cryptoendoliths. These are going to grow in spaces within rocks. A lot of times you get a crack in a rock. Many organisms can't survive there, but cryptoendoliths can. Think of these things living in underground water. It's hard to get to. Halophiles, they like salt, so they dig salt. What happens if you were to put, I don't know, human body in salt? It would float, not salt water, just salt, straight up salt. Yeah, it dehydrates. We used to take fish and throw it into salt just so we could dehydrate it, so that bacteria couldn't grow in it, because bacteria can't survive those hypersaline conditions. But archaeans can. Hyperthermophiles. Hyper meaning high, thermo, temperature. So these guys love temperatures between 80 and 122 degrees Celsius. What happens if you took any one of your cells and threw it into a pot that was boiling? Right? It just, the cell cooks. You denature every single enzyme. Once the enzymes are denatured, they're never coming back, the cell dies. Hypoliths. They live under rocks in cold deserts. We literally have an entire group of organisms that live in rocks in cold deserts. Lithoautotrophs. These are bacteria, so archaeans, whose sole source of carbon is carbon dioxide. So they pull carbon dioxide from the air, but they get the energy to process that carbon dioxide not from the sun, but by breaking down other rocks. Here you can see them in what are called mine tillings. Outside of mines, oftentimes you have very heavily polluted waters. Most organisms cannot live in that water, but archaeans can thrive in it because they're using the pollution in order to create their food. Metallotolerant. They're capable of tolerating very high levels of dissolved heavy metals, so copper, cadmium, arsenic, and zinc. I always love this picture, this exact picture. I can't find a citation for it, but this is one of my favorite pictures because they were going to test the metal content of that particular water source. And when they went to sample it, the bucket they were using dissolved completely. Yet, within that same water source, there are thriving colonies of archaeans. Osmophiles. They love sugar. You can, just like you can take salt and dry out a body, sugar can do it too. You can create salt mummies. In fact, I'm sorry, sugar mummies. That used to be a thing. Did you know, sidebar, one way, and I just found this out recently, one reason that mummies in Egypt there were so few mummies left is because people would eat them because it was basically like jerky. I know. Crazy. And it was mainly touristy people coming through. Also, ancient Egypt. I've been studying a lot of ancient Egypt lately. In ancient Egypt, there was a tourism trade and archaeology going on during the time of Cleopatra, because at that point, the pyramids were still so ancient that people would come from all over the world to see these. It was a big tourist attraction, too, just like it is today, except it was 2,500 years ago. The more you know. You can be a piezophile, an organism that lives optimally under high hydrostatic pressure. At the bottom of the ocean, at temperatures, that pressures that would absolutely crush you, these organisms thrive. Cryophiles or cryophiles, they like very, very low temperatures, like in permafrost. Xerophiles exist in desiccating conditions. Here, the Atacama Desert. Very, very low water. And then there's the polyextremophile. Poly means what? Multiple. They like multiple conditions, multiple extreme conditions. That they live in places nobody else wants to? Yeah, pretty much. If there's anywhere that can support that has a chemical that can be broken down, there will be life there that is breaking it down. It is likely, if we were to look at extraterrestrial environments, that any organism that we find on other planets, if they had a similar evolutionary path as on Earth, will look like the Archaeans. Mars, for instance, has regions in its deep subsurface that might have endolith communities or cryptoendolith communities. We know that life evolved on Earth. That doesn't mean that it can't evolve elsewhere. Probably an even more location would be on Jupiter's moon of Europa. Europa has basically a frozen surface with a sea underneath it. And in that sea, there are hydrothermal vents spewing out the same things that were spewed out on the Earth, at the bottom of the Earth sea for billions of years as well. If we're going to find something, we may find it extraterrestrial life, not as like an ET or something, but as an Archaean hanging out at a geothermal vent in our own solar system. Kind of